relates to clinical science as well. We have a clinical pharmacology branch that does pharmaceutical testing. But most of it is really basic science. Our strength is really in cardiovascular biology and immunology, if I were to try to choose where the majority of work is done. But we have quite a variety as well. In this particular department, this is the protein studies program. It was started by a gentleman named Dr. Jordan Tang, and you'll meet him later today. He came here as an undergraduate to get his degree at Oklahoma State University from Taiwan, and then went across the street for his graduate work and came to OMRF as a technician at that time, and then returned as a faculty member. He got involved in the interesting work looking at how the stomach digests protein. There's one particular enzyme there that is called pepsin, and a lot was known about it, but only in general senses. And he wanted to understand really the structure and the functioning of this thing. So he was involved in understanding what the biochemical structure, the amino acid sequence was of this, and that was quite a breakthrough at the time. And then he really focused on that particular enzyme for a number of years. When HIV happened, one of the first questions that had to get answered was, is this a virus? And when they found out that indeed it was a virus, they sequenced the genome of the virus, and most viruses have an enzyme as part of their genetic apparatus, and that particular enzyme was found to be the same family as pepsin. And all of a sudden, Jordan's phone started ringing off the hook, and everybody said, "Yeah! What was that you were telling us about aspartic proteases, and how do they work together, and what is their mechanism?" And so he very wisely changed his research program and started working on HIV. And one of the things that you do these days is to isolate the protein that you're studying and crystallize it, and then take X-ray pictures of that in such a way that you can see the atomic structure. Once you can see the three-dimensional structure of that protein, you can think of it sort of as a little machine, and you can think about building some sort of wrench that might fit into the active part of that protein and block it from acting. You can design inhibitors that will bind into the active site. In this case, this is a cutting enzyme. So we anthropomorphize a little bit and speak of it as a mouth. You can block that mouth with tightly binding inhibitors and then completely end that enzyme's ability to function. Therefore, if the virus requires that enzyme to be able to propagate, you just stop the virus from being able to grow. So that was the approach that he took, and they were able to crystallize, and they were able to design some inhibitors. And this was very exciting. While this was going on, he had a technician in the lab who wanted to go to graduate school himself and was interested in getting a project. And he thought he was one of these guys who was really into computers and what we now call bioinformatics. And he was analyzing protein structure on a computer. And he also knew that there was this is in the 90s now, the early 90s, he knew that there was this huge race going on called the Human Genome Sequencing Project. And he knew that there were a couple of, we now call them servers, but at that time they were mainframe computers, that he could go to overnight and search to look for new genetic downloads from the scientists who were working on his projects, and screen all of this new DNA to see if there's any evidence of other enzymes of this family that called aspartic proteases. And at that time we knew only about 2 or 3 of them, and he knew there must be others. And maybe we'll find genes for ones that are out there that we've never even seen before. So he would screen and screen and screen, and this was done long before Google was even a glimpse in anybody's mind. This was all done on command line through very primitive computer transfers, and I was working with him at the time, and it was really interesting to watch and see how he was doing what was really cutting edge stuff. And there was nothing, and then bam, he got one. Very exciting. And then nothing. And by the time, I think the summer that finished, and he had started this maybe 6 months before, he had 4 sequences. And he said, "Gee, maybe I better go to the boss and tell him what we've got here." And Jordan looked at him and said, "This is magnificent. What we have to do now is figure out where are aspartic proteases being expressed in the body. Where do they do their work? And the way you do that is a very standard technique involving a procedure called a North blot. And they set all of that up and did it. And what Gerald Kelch, the name of the student at this time, was able to show was that 2 of them were expressed in the brain. So he took that back to the boss. And as I understand it, Jordan practically picked him up and kissed him on both cheeks and said, "My boy, you really may have something here," because Jordan had been following the literature enough to know that there were suggestions that an aspartic protease might be involved in releasing the Alzheimer's peptide in the brain that goes out then and sort of coalesces with other copies of itself and forms these plaques and tangles that essentially goof up the neuron's ability to function and basically cause Alzheimer's disease. So if that were the case, then an aspartic protease were that releasing enzyme, then you could just sort of use that same technology to try to block that enzyme rather than HIV. And once again, Jordan changed his research program. And indeed, it turned out that that enzyme that Gerald discovered was, in fact, the very enzyme that does release the Alzheimer's peptide. Jordan calls it monopsin, and he has now spawned a startup company to develop inhibitors, and one of them is now in phase 2 clinical trials. And he tells me that he has even now continued the research with a synthetic chemist to design new and better inhibitors, and he's got one that's even better than the commercial one that he's working on trying to develop on the NIH-funded component of his laboratory. So the work goes on. I can show you some of the figures that came out of